So we will continue further. So in the last in our last lectures, we continued with the protein isolation, quantitation, and quantification and purification of it. So in order to do so, first we learned some techniques with the centrifugation. We can isolate the protein, and then we understood the quantification by Lowry and Bradford method, whereas mostly in the lab we use Bradford method. Now we will discuss about ultra sensitive fluorescence based protein quantitation. So in this biotechnique from 2003, we add a dye to the sample, we heat for 10 minutes, then we cool to room temperature and read the fluorescence. And in the protein quantitation, whatever the peaks that we see, the fluorescence, that will be corresponding to the protein microgram per ml. Then comes your protein purification, that is dialysis. So this is a technique used to remove small molecules from the protein solution or to change the buffer composition of the protein solution. So when we have smaller particles and bigger particles of a protein, and we wanted to separate them both, wanted to have a separation between both. So in order to do so, we use a dialysis bag which have our concentrated solution and buffer is outside. So at the start of dialysis, figure will be like in the left hand side. Then at equilibrium, all the small particles will come into the water and the bigger particles, that red one, will remain inside the bag. So let's see how we do this technique. Okay. So now comes the next part, the bigger part, that is your chromatography. So what is chromatography? If we see from the perspective of liquid chromatography, it's a term which refers to all chromatographic methods in which mobile phase is liquid and the stationary phase may be liquid or solid. So first one is the gel filtration chromatography. So in this, you put your protein sample and at the bottom is your molecular exclusion gel. So with the time, what you will see, there will be carbohydrate polymer heads, and there will be your molecules that the protein samples that you wanted to separate. So you can see the yellow one are the bigger one, green ones are the middle, and the red one are the smallest. Small molecules enter the aqueous spaces within the beads and they go down. Where the large molecules, they cannot enter to the beads, so they will flow down. So based on this size, from yellow to green to red, proteins will be separated. So same thing has been described in this diagram. This is how, like, this is HB, your mercury, Cephedex G75, Cephedex G10, and your protein is being separated in this gel filtration chromatography like this. So let's watch how gel filtration Cephedex G50 is done.
We're starting with gel filtration medium. This is Cephedex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the speaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1,000,000. Fluorescine, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescine, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first. to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
So this is how we make. This is how we make the dull exclusion chromatography. I hope this was clear. Now we go to the next one. Ion exchange chromatography. So basically in ion exchange chromatography, Protein in a solution has low ionic strength and is added to the column. So the protein interacts with the beads via electrostatic interaction. So here we have positively charged beads, positively charged proteins binding to the negatively charged beads, and the negatively charged beads will flow through it. So that's the basic principle behind your ion exchange chromatography. So this is the ionized form. This is the protonated form of one group attached to cellulose or agarose. So in an ion exchange, there are two types: an ion exchange and cation exchange. In ion exchange chromatography, matrix carries a net positive charge which interacts with the negatively charged protein strongly. While in the cation exchange chromatography, the matrix is negatively charged and it interacts with the positively charged proteins. So let's see how it is done.
Ion exchange chromatography is a chromatography process that allows the
Let me confirm if the signals are fine because I saw there was a low strength. If something goes off in between, please message me on the Viber. So we'll try to fix that because the stream seems to be poor now. In the afternoon, this happens to me mostly, but in the morning, it didn't went off. It just in the afternoon because of the higher traffic, it works like this. But let's see how it will work out. I hope, finger crossed, things doesn't stop.
Ion exchange chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into an anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. and at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both, protein B and C, carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column, as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained, and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified concentrated form.
So that was about ion exchange chromatography. Now I will take you to the next part, that is affinity chromatography. Mainly in this, it's based on the antibody thing. So here we can see that we load our protein, the red one and the blue one, and the column is filled with what do you say, the antibodies. So the antibodies, the red one will be the proteins will be recognized by the antibody, blue ones were not. So based on that, we will wash away and elute with the pH buffer 3. First, everything will come as a blue, then comes your red ones. So there are some drawbacks, that they bind to ligand antibody, and they are so strong that harsh conditions are needed to elute them. A non-specific binding may also occur, and this non-specific binding can overwhelm the correct binding sites at very high protein concentrations, leading to pure purification.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
Okay, so now comes the most important, that is the one of the most demanded chromatography that is highly used, is your HPLC, that is high performance liquid chromatography. So basically, we give a high pressure inside a column, C18 column, and we put our sample into that, and they get separated because of that pressure, and gives various peaks into the data collector, and we can differentiate that in a protein which proteins were present, mixture of proteins, and yeah, and we also got those proteins as separated. So in this high pressure liquid chromatography, it's an enhanced column method for protein separation. The resin size are more refined, and all the chromatographic methods described above can be applied with the help of HPLC column. And the equipment, but with the HPLC, the separation of peaks is almost always better than the standard chromatography columns. And the reason for this, that HPLC columns are packed with much smaller beads than used in the standard columns. So columns are quite large and tube containing small micromolar particles known as stationary phase, and the chromatographic bead bed is composed by the gel beads alone when they are inside the column. The sample is introduced into the injector and then carried into the column by following solvents. So you have absorbance at 220 nanometer with the time as 1, 2, 3, 4, 5 different values of absorbance. So let's check how we do practically the HPLC part.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In Hplc, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary action. In Hplc, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the Hplc instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Okay. Yes, Shrishti, I'm sharing all these PPTs with you with the video clips inside them. That's no problem. So, fast performance liquid chromatography is a type of liquid chromatography where the solvent velocity is controlled by pumps, and the pumps controls the constant flow rate of the solvents, and the solvents are assessed through tubing here, and the flow rate of the solvent is set through the computer input and controlled by the pumps. So gel electrophoresis, so protein separation, the most popular. So this we have discussed in details, so I will not go in that thing. We have discussed this STS page, various bands. Yeah, this one I want to discuss now from now here onwards. That is your isoelectric focusing gel electrophoresis. So in the isoelectric focusing gel electrophoresis, basically you have higher pH and low pH. Yeah. So the molecules that you will add into them, so they will be polyamphilites having multi-charged polymers. But when you put them into the high pH and low pH, they will find the pi value that is in which they are neutral and they can be get to that point. So pH gradient, polyamphilites with many pi values differ by 0.01. So in this isoelectric focusing electrophoresis, in IFE, the bands are even better. So this is unmodified version, and this is the modified, unmodified, and modified. In SDS page, we detect these charge per molecules, but in the IFA, we see a better charge. So let's see how we precipitate the protein for 2D gel electrophoresis, and then we will understand how we do this technique in exactly. So this video will not have voice, just follow the subtitles. Now, that was the first part, that is isoelectric focusing for the precipitation for the 2D gel. Now your all samples are being separated according to the pi value by the first dimension. Now we'll put this strip over the gel and we run the SDS page in order to identify a protein. So we will go for 2D electrophoresis with the red, blue, yellow, orange, and we have our SDS page and isoelectric focusing. So our gel will look like this. Our gel will look like this with these various proteins. All the proteome of a protein will be separated in this way. Now we will go with the how actually your how actually is your 2D gel electrophoresis practically done in the lab. So it will be shown by an expert professor in the lab for a long video. So I will recommend watch this video while making the notes, because this is one of the complex technique, but once you remember this technique, it's also extremely helpful. So all the techniques that you're learning here today, they have its own importance and they have its own role. I don't know when you will be using them in your life, but you will be prepared for them in advance. So that's the reason for this workshop. So I will share these videos first with you. In meantime, if you have any questions with me, you can ask me in the chat box. I will be happy to reply you. Otherwise, I will start sharing things with you. So maybe these videos I can share with you all, because sometimes they are not playing in the PPT. Mm. Okay, no questions so far.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has two sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip. On the negative side, you'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before the step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first, remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip. to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cipro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kumasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kumasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
Maybe I had shared all with you all videos now, so it will not, so you can watch them there finally. Maybe try to decrease the video streaming. I think it will be fine then. So you can watch these videos, students. I have sent you in the group. So all these videos have already been with you. So you can watch them later.